screen.
Globular, okay.
Globular. Because there are two types of protein, if you have learned, fibrous and globular, right?
Right, it's okay.
Two type of proteins are there basically. One is fibrous, one is globular. And fibrous is the one which is just fibrous in shape, fiber type in shape, and globular is something you can say circular, globule type, right?
Right, it's okay.
Okay. So fibrous are those which are helpful in giving the, you can say, they have the structural function. In the fibrous protein, they have the structural function. And globular protein, they are having the, you can say, not structural, but they are giving some important function to the body. Like if I say hemoglobin, so hemoglobin is also a globular protein, right?
Oh, wow, okay, okay, okay,
If
go on.
I talk about hemoglobin, so hemoglobin is a globular protein, right?
Okay.
So hemoglobin, it is a globular protein. And apart from this, if I talk about another protein, which I am saying is fibrous protein, in that case, I can give the example of like keratin, right?
Uh-huh. Sorry.
Okay, they
Yeah.
are present in our nails and in our hair.
Yeah, these are all proteins, okay.
So like collagen,
Oh yeah, okay, 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 okay.
right? So they are just having the shape of the fiber like this. So they are the, you can say, structural proteins. Basically, they are having the structural function, right? And these globular protein, they are having some internal functions inside the body, like hemoglobin, as you know what it does, right? Then
Okay.
you can say, what is the most important feature of enzyme? It speeds the chemical reaction. But what about enzyme itself? What happens after it have done the work in the reaction?
Uh, it just moves on. Nothing. See, it doesn't really, it breaks, it creates two products, but it doesn't really, it's not affected. It doesn't actually it
Yeah.
doesn't react. It just breaks them apart and then it's like still the same.
That
It just does the same.
And that means you can say it does not take part in the chemical reaction, right?
Okay, I'll keep that in mind. Doesn't take
Huh.
part in the chemical reaction.
Means
Okay, got it.
it won't alter its composition anymore, right?
Okay.
It won't alter its composition. It remains same. It functions, but it will help the speed to increase. It will not itself be involved inside the reaction to change its composition, right? And main important feature of enzyme is it reduces the activation energy of the reaction. And what do you mean by simple activation energy?
Uh, simple activation energy.
Hmm. Activation energy means it is the energy which is required by the reaction to take place, right?
Okay.
It
Okay.
is the energy which the reaction needs to happen to move further. For the reaction to take place, energy is needed, which is known as activation energy, right? Now see, if you see this graph, here we have the reaction. On the y-axis, we have the energy. Now, if you see the substrate, when they react, without the enzyme, the activation energy of the substrate is this much, see, right? But if we are having enzyme as a catalyst to help this reaction to speed up, so you will see the activation energy will decrease. Means this much amount of energy has been reduced, right? That means you can say it will lower the activation energy of the reaction.
Okay, understood.
Activation energy of the reaction. That means it will, the reaction will need lower amount of energy to take place if there is presence of enzyme, right? On
Okay.
what model does enzyme work?
Sorry?
On which model does enzyme work?
Uh, I want to say active site. I don't know.
That is lock and key model.
Oh, okay, okay. Lock and key. Sorry, sorry.
Or you can say lock and key hypothesis, right?
Okay.
And what it does, what is the meaning of lock and key hypothesis?
It's basically like it has a specific shape for specific substrates. Yeah. It won't really have that effect with different, substrates, only specific ones, right? A
And
specific 3D shape, right?
what do you call this thing?
Active site.
And what is active site?
The specific shape, I don't know.
Active site is the specific shape on the surface of the enzyme to which the substrate binds. right?
Okay.
It is a shape, it is a groove on the surface of the enzyme to which the substrate binds for its activity, right? And when it binds, see, when it will bind to this shape, it will just form an enzyme substrate complex. The enzyme will bind with the substrate, and we will call this figure together as enzyme substrate complex. And further, this enzyme will help this substrate to break down into products. Now see, products are released. After that, these products are released and enzyme will move on. It will not go together with the products. It will remain there. It will wait for another substrates to come and join to the active site. That means I can say enzyme is substrate specific. Am I correct?
Substrate specific. Okay.
Means
Got it. Substrate specific. I have to keep that in mind.
This is an important term while explaining enzymes. That means a particular substrate can fit in the particular groove or particular active site of the enzyme. If the substrate will change its shape, it will no longer fit in the enzyme. So that means particular enzyme have a particular, you can say it is complementary to the shape of the substrate.
Understood. Okay.
Okay. So enzymes, not all the enzymes will bind to all the substrate. No, only few enzymes are marked for few substrates. There are different enzymes for different substrates, right? So they are substrate specific, you can say. So this is a lock and key model. See, this is the shape of the active site, and see, this is meant for this substrate only. If you are having some substrate like this, so it won't fit here. So it is substrate specific. Now see, when it will bind here, it will form the complex together with the enzyme and substrate. It will be called enzyme substrate complex, or rather you can write it like this also. Right? And then you will see the activity of enzyme will help this substrate to break down into the products, and these products will be released. This is the lock and key model. However, a little modification was done in this model, and after that, a new theory was given. What was that?
I really cannot tell you. Oh, it's written here now.
It is induced fit hypothesis. Have you heard it? It is there in your syllabus, I think so.
Let me see. I really don't know anything about enzymes, honestly.
Okay.
I'm really weak in this or many of these topics. How do I say? Yeah.
You like biology?
I really like biology, but my knowledge is weak because over the years with multiple subjects, I really just didn't focus on bio much. And that really, it doesn't really help.
Bio is an interesting subject.
It is really, honestly, of all the sciences, I love this the most.
So no problem. Little modification was done in the lock and key theory, and then the theory appeared, which was called induced fit hypothesis, right? What happened in this theory is, as the name tells you, it is a modified version of lock and key theory. What is the modified version here? Here, the modification says that the induced fit model is modified in what sense? That it has the tendency, there is a tendency of the active site in the enzyme to change the shape according to the substrate. Getting my point?
I'm getting your point, yes.
I mean, this theory is little modified. In what sense? That the enzyme has the capability to change the shape of the active site according to the substrate, so that the substrate can fit inside this enzyme. There is little distortion in the shape, but it will take the shape so that this substrate may fit inside the body of the enzyme.
Got it.
Like, see, it is circular, it is square shape, but it won't fit here, it won't fit here. Now see, in this figure, you will see the shape of the active site is made in such a way that this substrate has easily adjusted inside the body of the enzyme. And now it will function as we know. It will just form an enzyme substrate complex, and then it will break down into products, right? So little, you can say a minor modification is there. And hence this theory came into, you can say, agreement. Why? Because this was a little flexible theory, right? It was not that rigid. There was little flexibility in the shape of the enzyme, so that the substrate might easily fit inside the body of the enzyme. This was induced with model or hypothesis. Now, what are the factors on which